So objective of my study was to assess the activity of antioxidant enzyme caused by PVC microplastic and to determine the abnormalities and chromosomal damage caused by PVC microplastic. So after finalizing, the PVC microplastic was procured from Palghar, Mumbai, Maharashtra, and then it is characterized by using stereo microscope, Raman spectroscopy, uh, using same image technique, and microplastic size analyzer. This all pictures show the microplastic which are chosen for exposure are of different size and shape, less than 5 millimeter in length. Uh, so this was my model organism. Model organism was basically the Oreochromos mosaicus, commonly known as tilapia. It is a well-known organism for the toxicity studies, and the doses were finalized based on the LC50 dose, that is lethal concentration dose, and based on that, three final doses were finalized, and these doses were induced for the further studies. So basically, the fishes were exposed with PVC microplastic, and then it was dissected and different organs were removed. And after removing the organs, the further test like toxicity test and biochemical test was performed. So basically, the biochemical test which were performed was catalase test, superoxide dismutase, ALT, and ALP test, and genotoxicity test was micronucleus test, and some more test was performed beside this test. So biochemical test, the result of the chemical test shows there was significant increase in the catalase and SOD activity with increase in the concentration of PVC microplastic in gut and liver tissue. The TBAS and GSS shows TBAS and GSS showed opposite trend where TBAS showed that with increase in the concentration, the enzyme activity also increased, but GSS does not show such trend. LP and LT basically the liver function test, and it shows with increase in the concentration of microplastic, the level of ALP and LT also increased. Protein and carbohydrate shows opposite trend where it was noticed that with increase in the concentration of PVC microplastic, there was decrease in the total protein and total carbohydrate. Because of the stress, the protein and carbohydrate was utilized at very high extent. So this was the micronucleus test. Micronucleus test basically shows the chromosomal damage. So if many micronucleus are dead, then it shows that chromosomal damage is very high. So I'll show you the with increase in the concentration of microplastic, the micronucleus also increased. So at highest concentration, that is 45.04 mg per liter, there were many micronucleus in one slide. That showed the chromosomal damage was very high at higher concentration. Uh, the last was the gene expression test. So gene expression test was done on CYPA gene. CYPA gene is nothing but a gene which is highly expressed if the environment is very toxic. So it was saying that with increase in the concentration, the CT value was decreasing. So the significance of CT value is that if the CT value is high, that means the toxicity is very high. So at highest concentration, that is 45.04 mg, the CT value was less. That signifies the gene was highly expressed at highest concentration. Okay. Control 1 minute. Okay, done. So my conclusion is there was increase in the catalase and TBAS activity which show there was oxidative stress in the fish after exposure with the PVC microplastic. Then LP, LT and SOD show the liver damage, and chromosomal damage was noticed by elevated gene expression and micronucleus activity. So future scope, this all microplastic was single studied, so we can study this microplastic with the other environmental stressors, and then we can give a better picture of it. And more molecular study is required in this study. And this is all about my study. Thank you.
Thank you. So maybe just one question because these are all oral presentations. If anybody wants to ask, maybe we'll allow one question. If anybody. Thank you. Thank you. So invite our guest speaker, Atul Kumar. Atul Kumar. Yeah. Oh. So you next persons be ready. So immediately have to come to the dais. Raj, okay? Then other then I mentioned already the names. You please be ready to come to.
Good afternoon, everyone. I am Arun Kumar, PhD scholar from National Dairy Research Institute. My presentation title is an in situ produced vitamin B12 rich ready to reconstitute nutrients. Vitamin B12 is an essential vitamin which is predominantly present in only in animal source, which is lack in plant source. Our motto is to synthesis biosynthesis vitamin B12 using the plant source components. And mainly the commercial supplement cyanocobalamin is a synthetic form which may create concerns related to the cyanide release during the metabolism. Who have the kidney related functions or liver related issues, they may can't bioavailable the vitamin B12 related to the cyanocobalamin. And another issue, the natural forms have more bioavailability compared to the synthetic form cyanocobalamin. And our India, 49% of people are deficient in vitamin B12. Especially, we have around 29% of vegetarian population in our country. And if we fermented the plant sources and milk waste sources such as whey powder and soya powder and sorghum powder, we using the base fermentation matrix for synthesizing vitamin B12. And we took these three components at tree level and we optimized by using the vitamin B12 synthesis, biosynthesis yield. In that our observation, around the 10% soya production, 4% sorghum and 3% whey powder, when we combine with the biofermentation by propanobacterium, we observed around 4 microgram per 100 ml for 100 ml of the media. And another, we optimize the fermentation condition, especially the incubation period and the mm, culture propagation percentage. In the observation, we optimized around 10 milligram per 100 ml of Propionibacterium shermani culture, yielded the for 4.5 microgram of vitamin B12. And when we go for the fermentation condition, 72 hours of anaerobic condition, 48 hours of aerobic condition, yielded 4.7 microgram vitamin B12 per 100 ml of the media. And this is the final output of the in situ production of active vitamin B12. We are taking the sorghum, soya powder, and whey powder related to the optimized concentration, and we are homogenizing the mix, and we autoclave by sterilizing free from other microorganisms, and inoculation using Propagnatum shermania, the culture we isolated from the cheese, and we are incubating and the 72 hour of anaerobic, 48 hour of aerobic condition, and we are choose the spray drying process as we can commercialize easily to the industries, and we produce the vitamin B12 rich fermented base. And for the Nutrimix, we are planning for the commercialization as such as Horlicks, Boost, such type of station product. For the Nutrimix powder, we took other some flavor enhancement and the commercial flavor enhancement and color appearance. We took the cereal malt and the cocoa powder and sugar, and we optimized the concentration 50% of dried fermentation base with 12% of cereal malt and 30% of sugar and 8% of cocoa powder gave the better dispersibility, better wettability properties, and the overall acceptability in the sensory evaluation. And the characteristics we evaluated in the nutritional composition, the Nutrimix providing 72% of carbohydrates, 18% of protein, and the 3% of fat content. And in the microbial property, it showed it under safe and FSSAI standard. The spore count and the yeast and mold count was observed, and the standard plate count around 4.57 log 10. And the sensory appellation and the 9-point hedonic scale, it showed around more than 8 the in color appearance and sensory ability and the overall acceptability. And the color parameters, it showed the L value and B value around the positive. It represents the powder color appearance will be slight yellowish to white color. This is a final conclusion. The chromatogram of vitamin B12. The developed vitamin B12 around 20 gram of serving, providing 100% RDA recommended by the ISMR. 2.4 microgram it is providing. It is giving more than 2.6 microgram. And I am concluding this presentation by acknowledging the ISMR for funding of this research. Thank you.
Thank you. Just one my clarification.
Yes.
In the final, the product, dried product.
Yes, sir.
What is the concentration of vitamin B12?
Sir, vitamin B12 will be 2.6 microgram per 20 gram of powder.
Oh.
Like if we are taking 20 gram of serving, we will make 100% RDF vitamin B12.
Any from? There was just one question. There are no questions. Thank you.
Thank you, sir.
I will invite Sandeep Kumar. Next presentation.
Good afternoon to all of you. My name is Sandeep Kumar from Indian Institute of Technology, Roorkee. I am presenting here locating the disease hotspot in tea field using the geodata area university. This is the content of today's presentation. Coming to in particular India, the industry facing to difficulty to controlling mosquito bug in tea field using the pesticide spray. In a traditional method, ground sampling and visual interpretation are time consuming and level intensive. So the alternative option is remote sensing, overcome this limitation and enable the pesticide monitoring and damage estimation of leaf. Combined in the field data with remote sensing also improve the productivity daily. Now they don't provide a real-time data collection for pest life cycle management. I have I have collected the drone data of multi-spectral and RGB measure and combining with the new technology of clustering. Clustering is the combine the coordinate with the convert into the cluster and collect the digit hotspot in the tea field for actual and hotspot of the tea field and spraying the pesticides. I have using 4 type of clustering algorithms: K-mean, KN, DBA scan, and optics. My objective of this presentation clustering the geospatial coordinate and evaluating the accuracy of the 4 types of clustering algorithm and developing the hybrid approach of the clustering. This is the study area of my presentation. I have collected the data from the Valparai district of the Valparai city of the Coimbatore district from the Tamil Nadu state. I am coming to the methodology section. I have collected the UAV RGB measure from the tea field
And after that, we use the machine learning and deep learning and AI algorithm to detect the disease, and after that, segmented the whole image into small section and extracting the latitude and longitude coordinate of the each section of the image. And after that, using the clustering technology for the uh, for estimation of the disease hotspot. And after that, applying the pesticide, using the decision support system for applying the pesticides. I have collected the data of RGB image and extracting the latitude and longitude of that image, and using the four type of clustering. In the K mix, I have used two types of input parameter: number of cluster and then random distance. In the KNN, TBS scan and optics, I have used two types of parameter. One is the effective radius, and other is the Haversine, using Haversine formula for estimation of distance between the two coordinates. I have using the Haversine formula in place of Euclidean geometry, because the Haversine formula calculates the social distance between the 2 points on the curved surface of the Earth. So I have used this formula. I have using the Python language for the developing the hybrid approach in place of K-mean and KN. I have merged the K-mean and KN for the estimating the disease hotspot in the deep fields. I have using two metrics indexes for the evaluating the models. The first one is a Silhouette index that is vary from -1 to +1, and also there is Bloodline index for the evaluating the intercluster and interclusters. I am coming to the real section, the result section. We have found that hybrid KNMs are best Silhouette index and the best value index. I am also I have collected the different type of cluster, number of cluster by the different method. But in the case of optics method, I have getting a very huge number of clusters. So that is not quite effective. So I have used the K-means and K for the quite effective. And this is the output of the result of the previous tables of the input data set of the latitude and longitude. I am coming to conclusion part. I have used the I have using the hybrid approach for the best estimation of the disease hotspot and in a deep field. Also getting the highest Silhouette index 0.83 and the Bloodline index. Then this is my plan. Thank you.
Thank you, Sandeep. If there are any questions from audience, of course, your paper is full of mathematics and statistics. Okay, so equations and all. Okay, thank you. Thank you. Next presentation is by Hemraj. Hemraj Kamanath. Kamanath is not here. Then Kadagi Ravi Kumara, please come. So select your presentation.
29, 20. 13, they have such 40. Ravi Kumar. 29 name, just a number.
Good afternoon, all. I am Ravi Kumar. Today I am going to present the HRLCMS profiling of phytochemical constituents of Bignonia magnifica and evaluation of antimicrobial, antioxidant, anti-inflammatory, and anti-cancer potential. So, introduction. Nature has given us rich biodiversity and wide range of plant species are grown in different parts of the world. So the World Health Organization estimated that 80% of the people in developing countries rely on traditional plant-based medicine for their primary healthcare needs. Bignonia magnifica belongs to the family Bignoniaceae. So it's an ornamental plant. It stands as a novel underexplored medicinal plant. So this is a materials and methods. So this Bignonia magnifica was collected from the University of Mysore Manasa Gangotri campus. Firstly, leaf powder was filled in the oxalate extraction apparatus and then added the polar and non-polar solvents like ethanol, methanol, ethyl acetate and chloroform. So it starts to boiling. It can leach out all the secondary metabolites from the sample. It is called as crude extract. Using this crude extract, we can go for the biological activities and then go for the phytochemical analysis and then isolation and characterization of bioactive compounds. So the results, authentication of plant material from the Botanical Survey of India, Coimbatore, Tamil Nadu. So antimicrobial activity of Bignonia magnifica against gram-positive, gram-negative bacterial strains and fungal strains by well distribution method. So this figure shows the good antibacterial, antifungal activity. So antioxidant activity of Bignonia magnifica was evaluated based on the ability to scavenge stable DBPH and ABTS radicals. So in the ethyl acetate extract, it shows the 98.97% of PPPS scavenging. So in case of 91.06% of ABTS scavenging, as compared to the standard gallic acid, it shows the 89.15% of DBPS scavenging. In case of ascorbic acid, it shows the 75.1191% of ABTS scavenging. So it shows good antioxidant activity. So anti-inflammatory activity of Bignonia magnifica leaf extract shows by heat and hypotonic induced hemolysis, it shows the moderate anti-inflammatory activity. So high resolution liquid chromatography mass spectrometry, the work has been done from SAIF at IIT Bombay, Mumbai. So this chromatogram shows the separation of these peaks represent the separation of the compound, height of the peaks represent the abundance of the compounds. So these are all the bioactive compounds are present like phenidinone, alamandine, pentamethoxyflavone, simifigine, tocopherol, hydroxymugenic acid, and phytospingosine, scleracarastitin, icosanidol, lucidin, cassidem. These are all the bioactive compounds present in the sample. And coming to the point, anti-cancer activity against a Hep2 cell or liver cell line. So these cells shows the at an untreated cell, these cells shows the cell proliferated. In a treated cell, it shows the cells are suppressed or cells are disintegrated. So as concentration increases, cell viability also decreases, and it shows high cytotoxicity and gives the IC50 values to 210 microgram per ml. And coming to the point conclusion, in this study we have demonstrated for the first time the ethyl acetate extract shows the good antioxidant, anti-inflammatory, antimicrobial, and anti-cancer activities as compared to the chloroform, ethanol, methanol extract. This data justified the folkloric medicinal value of this plant to treat various diseases. The present work can be considered a prelude for exploring novel bioactive compounds to control problematic microbes and excited free radicals. Moreover, these anti-cancer property of this plant to treat cancer. Overall, the result provide a groundwork for further studies focusing on drug formulation as a natural remedy. So these are all the references. Thank you.
Okay. Thank you. So, if any question?
Why did you choose this exotic plant?
Uh, it shows the good biological activities, sir. It is a medicinal property. That's why I have to.
But we should exploit our own wealth that we have. Why do you choose?
Yeah, there is
There are many ignoracy plants in India.
work is not done at sir. That's why I have to select him. And it also shows good antioxidant activity for to control some of the cancer also. That's why I have to select this cancer. What's Professor Mesha sir?
Thank you.
Thank
Next presentation by Karthik Prakash. Then after this presentation, the next presentation would be by Eridora Kaifoza from the National University of Uzbekistan. Next, after this.
Next.
No, next. After this, right?
It's very.
Because you'll be ready. I told you to be ready.
Greetings everyone, I'm Karthik Prakash from Sati University of Science and Technology, Chennai. I would like to present my work titled Marine Derived Bacillus Party from India's West Coast, multifunctional probiotic candidate for sustainable agriculture against antimicrobial disease. As we know, the aquaculture is an important industry worldwide, currently providing more than 50% of the seafood production in the world. And as you can see, the producers for around 70% of the aquaculture producers are from India and China, Indonesia, Vietnam, which belong to the Asian continent. And as the aquaculture is growing, the demand for the seafood is increasing, and it increases the stocking demand in the fishes, and that will eventually increase the water quality, decrease the water quality, and it will cause bacterial pathogens. As you can see, the bacterial pathogens such as Aromonas hydrophilia, Yersinia rickerii, Vibrios cause millions of dollars loss in bacterial mortality in fishes. And for historically from nine. Okay. Okay. To combat the AMR caused by the antimicrobial use in agriculture, we came with an hypothesis to isolate a bacteria from an existing fish. We took three fishes, economically important fishes from the west coast of India: Indian mackerel, oil sardine, and squid. And we isolated the gut-associated bacteria. As you can see, we collected around 177 morphologically distinct bacteria, and we didn't see any growth in the TCPS agar, which is a selective media for Vibrio species. And no, just yeah, there are several slides. Huh? No. Can you scroll down? Can you scroll more? Okay. With this, we isolated three bacteria, namely IM30 from Indian mackerel and IM23 from Indian mackerel, OS40 from oil sardine. And in the safety assessment of the bacteria, we took hemolysis pattern, but these isolates from Indian mackerel exhibited alpha hemolysis, and OS40 produced gamma hemolysis, which is safer for the living species. And for the antibiotics susceptibility test. Three bacteria were sensitive to commonly used antibiotics across India, and I can tell that these bacteria are safer for human consumption. Yeah. Okay, I'll just come to the conclusion. And after sequencing, we came to know that OS40 is Bacillus subtilis, and it was anti-hemolytic, and it produced this like basic resins like inhibitory substances, which is an essential trait of the bacteria species Bacillus subtilis. And these OS40 can produce biofilm, which will be necessary in colonizing the fish gut once it is introduced. And that's how my work has concluded, and we'll proceed it further through in vivo trials in fishes. Thank you.
So you are from which university?
I'm from Satyam,
Pratyabhama University. Where? Maharashtra.
Chennai.
Chennai. Okay, thank you. So yeah, next presentation by Nidaria, Eridova, Kyle University. Okay. So from National University of Uzbekistan. Okay. 5 minutes. Huh?
For next. Good afternoon, everyone. My name is Isidora. I am from Uzbekistan. My presentation is about the effects of plant growth, promotion, bacillus in the fetus, and mycorrhizal fungi on the growth and physiological properties of ginger. We have a little time, so I will show main tasks. Plant growth promotion traits, bacterial in the fetus, and showed several beneficial traits such as phosphate solubilization and EAA production in the means and enzyme activities, indicating its strong potential as a growth promotion bacteria. And plant growth results, both in the feeds and microbiological fungi, improved plant heat, leaf number, leaf length, and leaf width compared to the control. However, the co-inculation treatment produced the highest and most significant increase in all growth parameters. Physiological properties, the combined influence also engaged chlorophyll A, chlorophyll B, total chlorophylls, and carotenoid content, showing improvements in physiological performance. Relative water content increased with both inoculants, and the highest value was reported in the combined treatment and indicating improved water status of the plant. Soil chemical properties. Both treatments improve total phosphorus, nitrogen, and carbon content in the soil. The co-inculation treatment resulted in the highest nutrient levels, demonstrating better soil fertility. Yeah. Soil enzyme activities. Soil enzyme activities such as catalyze, invertase, urease, and phosphates significantly increased in all included treatments. The combined treatment again showed the highest enzyme activities, indicating improved soil biology health. And our conclusion is both endophytes and microbial fungi individually improve ginger plant growth, physiological traits, soil nutrients, and soil enzyme activities. However, the co-inclusion of bacillus endophytes and mycorrhizal fungi produces the strongest positive effects. Therefore, using these microorganisms together can be considered an effective and environmentally friendly approach to promote plant growth and soil quality. Thank you.
Thank you. So that comes all the backlog. Clearance of the backlog, yesterday's backlog, and we start with today's schedule as listed here. That's right. Alakananda is here. Yeah, please come. Then next person, Pankaj, right? Pankaj Chauhan is here? No. No. Oh, I get it for a wrong one. Dharmesh is here? Okay. Here it is. Next person. Four minutes for presentation.
Sure.
1 minute for any discussion.
Good afternoon. I'm Alakananda. I'm from the Department of Computer Science from National College, Baswambudi. My topic is on real-time climatic changes, which is a study for sustainable development. It is basically a computer science-oriented subject where we have tried to. Next slide. We are trying to study the climatic changes through a predictive model of machine learning. Here we are using layers of approach. The layers are like this. We have the ground layer where we are talking about IoT devices which are responsible in collecting data such as temperature, humidity and various other sensor devices which are sending the data to the imageries which are basically EOS, Earth Observatory Systems, which are collecting the mapping devices and sending the predictive analysis to machine learning algorithms. We have adopted few algorithms for which we study and predict the model. Thereby, we can also have a proactiveness where we can predict how fast any changes are occurring. The decision support system can be used, and also the machine learning techniques such as regression model, CNN, and various others can also be implemented. Just these are my objectives. As already existing, the problem statements have stated that already some few models are existing, but it was not integrated. Unified was not there. I tried to bring in from multiple sources this data and try to build up the layers and extract the data and finally make a decision support system in order to make a decision for the changes which are accredited. Next. This is the methodology of the framework which I have given. There are various applications. Few of them we have taken, that is sustainable agriculture, clean water access, and renewable energies. This is my conclusion. I would say that with this, and since the study is still going on, it is in the process. We are now trying to put up some machine learning techniques which are very recently done based on quantum. So it's called quantum machine learning techniques. The CNN approaches we are using. This is where we stand. Thank you very much.
Thank you. Thank you for your nice presentation, sharp presentation.
Yeah, I made it very big,
Yes,
sir,
yes.
because the subject is very vast. It takes layers of approach. With whatever possible time, I have tried to conclude. Any questions, sir? Thank you very much.
Thank you. So, Hello? Sir, Dharmesh, yeah.
Yeah. So good afternoon to all. This is Dharmesh here from Dr. Subhash University, Junagadh. Today's my topic, that is the integrated effect of the PGPR consortium on the growth and yield of the wheat, particularly in the Soras region. So the problem which is inspired, that is the uncontrolled use of the chemical fertilizer, which loses the natural fertility of the soil. Now we know all about the PGPR. They are the plant growth promoters. There are the different traits which improve the plant growth as well as the yield. How it is be beneficial? There are several the mechanisms they have through which they improve the plant growth and yield. They increase the ethylene level, zinc, phosphorus stabilization, as well as the indoleacetic acid, which is one kind of growth hormone. So till today, there are lots of so many n number of the organism which has been isolated and identified, which having the PGPR activity. Now, my topic is why I choose the consortium, because each and individual organism that doesn't have the all possible traits. So we need to combine that whole organism in a single solutions, so that can be utilized in place of the single one. Which methodology I use? I select the sampling area, specifically from the Saurashtra region. Three districts I choose: Jamnagar, Junagadh, and Rajkot. Here, how I collect the rhizospheric soil. For that, I choose the 5 healthy plants in between the 15 to 30 days. Majority of the isoregion, which I was isolated on the neutral agar media and Pseudomonas agar media. Then after I select them based on their morphological features. Especially I select the different traits, like 5 different traits, like acetic acid, phosphate, zinc, ammonification, as well as the hydrogen cyanide. Along with that, I also test for the seed germination assay and calculate the vigor index. These are the same factors. Now the question is arrived that why and how I select the potential isolates. That is basically based on the phosphate, zinc, and acetic acid plus germination assay. Using this, I select the 7 most efficient isolates. Finally, I do the genomic DNA isolation and 16S amplification for their molecular identification. As we know, for the preparation of the consortia, we need to check the compatibility. The field trials I do, that is control T1 and T2. In the T1, I use the consortia plus Acinetobacter. In T2, that is only the PGPR consortium. The trials and treatment which I used. And these are the different parameters which are I studied during the field trial and statistical analysis. Now directly move to the result. These are my isolates and the phosphorus solubilizations which I observed. Solubilization index I observed between the 7.92 to 3.28. And these are the pictures. Solubilization index which I observed majorly in the 7GS. Okay, zinc solubilization you can see here, and the pictures. Indoleacetic acid productions. A few of them are very, as well as the germination assay, you can see the pictures. Combine this all 7 isolates. Basically, these are the final table into from which I decide their features. Actually, I choose that 7 or 6 to 7 features. Based on that, I decide this 7 isolates. Identify them by the NCBI submission, phylogenetic tree. The compatibility test I checked on the nutrient agar plate by just overstreaking them. These are the carbohydrate utilization and the field trial data. The box plot is clearly says that the T1 is highly, you can say it's improved the chlorophyll content.
Here, same way, you can see here the tiller number and the shoot length is also improved in the 45 days as well as the 75 days. These are the pictures, shoot weight, and their box plots. Okay. So should I go for the conclusion finally? Majority of the isolates are belongs to the Pseudomonas, Iridia, and Acinetobacter. I say that 29% of the respiratory bacteria are the beneficial one, and we can use this consortium as a fertilizer industry. The one of the most the question which is the long-term preservations that we are not actually able to resolve. The co-inoculation of Acinetobacter coecum plus PGPR consortia with the cow manure, fertile agriculture soil will improve. It gives the better result than the that chemical fertilizer fertified soils. Thank you so much.
Thank you. Any questions? Yeah, please.
Did you re-isolate that in the
Okay, after the treatment, okay, I have not checked the whether it is there or not.
whether same kind of bacteria down with
Actually, it's difficult to identify, but we can do that. But I have not did that process.
So it's an easy method, no isolation, same way whether we need.
Isolation is possible, but for that we need to go for the identification of the same organism.
Like a morphological identification.
That is possible. That's possible. But I have not did that. I can do in the next trial. There is enough. It's only a single trial.
Whether it
We'll
is
go for the second.
supportive data.
Yeah, definitely. Thank you.
Thank you.
Any more questions?
No, yeah. So next presentation by Ms. Pooja. You're here. Okay, yeah, please.
Good afternoon to everybody present here. So my name is Pooja and I'm from Maharashtra. And today my presentation topic is the scenario of sericulture and the problems which are faced by the farmers in Umarkhed Taluka, which is located in Maharashtra in Yavatmal district. So actually, as we know that what is the sericulture means the rearing of the silkworm is done there. So I don't want to just explain this. You all know very well known that about the sericulture and the rearing of the silkworms. So many of the insects are there which produce the cocoons, and that likewise the silkworm is also which produces the cocoon. So these are the various types of silkworms and the silks which are produced by the worms. In Maharashtra, mainly the mulberry and the tussar silk is produces in the much amount. That's why for the mulberry plant, it is known as the tutti plant. Means in Maharashtra, commonly it is said as the tutti lagad. Okay. So material and methods are there. I studied in the Yavatmal district, which is the large area of that district is present. And the secondary data was collected from the sericulture office. So this is the map of that taluka. In the study, the questionnaires was taken from the farmers, and the visits are where there in the sericulture plants. So in that, the various questions were also there. Likewise, the education, the is there the participation of the woman in the sericulture, or what type of income is they are getting from the sericulture. So these type of questions are there in the questionnaire form. So actually, why sericulture? We know that it is the this low investment crop. The high returns are there, and it is also economical friendly farming. Well, as we know that. Okay, so In this chart, we can see that in the Umathed Taluka, there is the production is too much in this district. So, as there is the production is high, but then also there are the various problems which are faced by the farmers in the taluka. That is, miss the main problem is that of uji fly, and then again while interacting with farmers, it knows that in the starting years the production is high, but when as the after some years the production is get decreases, and due to the lack of techniques there is also a low production in the taluka. So and extremely because of the seasonal conditions, it also produces the disease worms. So, and the major problem is that the market. Actually, from Maharashtra, the many much of farmers came to the Ramanagar, as you know that it is nearest to the Bangalore. So that is the market problem is the main problem which is faced by the farmers. And again, the quality of the seeds means actually while interacting with farmers, it comes to know that means the seeds which are provided by the government they are not of good quality. So this is also a major problem again. And because of the lack of education and the technologies, the farmers face this the low production problem. So this was the field visit and the questionnaire survey which was done by me in the sericulture farms. And the challenges or opportunities are there. The trainings or the motivations are needed. And the market facilities may be provided in the sufficient way. So, thank you. These are the references. Thank you.
Thank you. Any questions from the audience? Thank you, Pooja.
Thank you.
So, yeah, we will break for lunch. Now it's almost 1:45. Half an hour lunch break. That is 2:15. Please join.